Hello, class. My name is Joey Kuzmic.
And my name is Kyle Brown, and today we will be discussing mitigations to climate change in terms of bacterial carbon sequestration. The year is 2050. Ecological chaos runs rampant throughout the world as extreme weather patterns, land degradation, rising sea levels, and pollution have claimed the survival of several million species. In geological time, this mass ecological destruction has occurred not as a long, gradual, expected process with many culprits. It has actually transpired as one of the largest and fastest genocides ever at the hands of one species, Homo sapiens. One recently evolved species has managed to destroy a world which has survived normally for billions of years only for the betterment and progress of its own members. If humans continue to live this way in the epoch known as the Anthropocene, which is characterized by human actions causing permanent global changes and catastrophes, this dystopian world I just described in the world 2050 could very well become a reality. One of the largest contributors to global climate change stems from the increased release of greenhouse gases into the atmosphere. One of the most important of these greenhouse gases in terms of its effect is carbon dioxide, which absorbs infrared radiation from the sun and traps the sun's heat in the atmosphere, as depicted in the picture on the bottom right of your screen. A sustained increase in the concentration of CO2 in the atmosphere can be attributed to many anthropogenic activities such as changes in land use, deforestation, industrialization, transportation, and modern lifestyles. Specifically, CO2 has increased approximately 43% since the Industrial Revolution and is expected to further increase to 60% in 2100 if the current trend continues. For this reason, mitigation strategies are one particular interest to both ecological community and to humanity as a whole. In order to mitigate the effects of climate change in terms of CO2, scientists have created three primary practices. One, renewable energy and proper fossil fuel utilization. Two, carbon sequestration. And three, the use of unconventional fuels such as biohydrocarbon and biodiesel. Today, we will be focusing on CO2 sequestration when specifically applied to bacteria. The primary literature source which we have chosen to present on is intercellular carbonic anhydrase from Cytobacter fridui and its role in biosequestration, which can be viewed in the top right picture here. Carbon sequestration refers to the capture and secure storage of carbon that would otherwise be emitted to or remain in the atmosphere. One of the most effective methods of biological carbon sequestration involves the use of microorganisms such as bacteria that are able to take up atmospheric carbon dioxide and enzymatically convert it to some other storage form. One of the key enzymes in this process the researchers have identified is carbonic anhydrase. Carbonic anhydrase is one of the fastest known enzymes able to hydrate CO2 to biocarbonate and then convert this biocarbonate into calcium carbonate, which is a solid mineral produced by carbon sequestration. This process is described in the diagram to the bottom right. As it shows, carbon dioxide from the atmosphere is taken up and in the presence of water, converted to H2CO3. It is then converted to bicarbonate and eventually will form calcium carbonate in the presence of calcium ions. The research from this article decided to study the carbonic anhydrase activity of bacteria taken from soil samples in the Himalayas. They decided to obtain soil samples from the Kumin region of the Indian Himalaya because it is one of the richest bioresources of unique microorganisms.
So, for the materials and methods, first they needed to test the soil samples they collected to see if there were any bacteria in them that even had carbonic anhydrase activity. The soil samples that they collected and shipped from the Himalayas were then crushed into fine pieces, labeled, and dissolved in saline water. In order to isolate any carbonic anhydrase producing bacteria, the researchers performed a plate assay in nutrient agar containing a compound called nitrophenyl acetate. The production of a yellow color with this nitrophenyl acetate was a sign that the bacteria on the agar had carbonic anhydrase activity. So, to put that in much more simpler terms, they took the solution containing the soil and grew it on a specific gelatin substance to cultivate the bacteria in the soil that had carbonic anhydrase activity. And this is basically what the pictures on the right are showing. where the yellow color produced on these agar gels is bacteria that has carbonic anhydrase activity. So from there, the colonies of bacteria that produced the yellow color were then isolated using the streak plate technique. After streak plating, they analyzed 6 randomly selected colonies, and a single colony was labeled as having higher carbonic anhydrase activity than the rest of the bacterial colonies that were tested. So to continue, in order to test the carbonic anhydrase activity, the researchers needed both purified and crude forms of this enzyme, not just the bacterial colonies themselves. A purified form is just a single homogeneous protein mixture, which will show the highest enzyme activity. And a crude form is an impure or heterogeneous enzyme mixture with other proteins in it. And this type of crude form will exhibit less enzyme activity. In order to actually gain access to the proteins, the bacterial cells needed to be lysed or burst open. After lysing and then using centrifugation, the remaining supernatant, which is the liquid mixture above the solid residue containing the crude enzyme extract, was further purified through a complex process of loading, washing, and eluting the extract, producing the homogeneous form of carbonic anhydrase specific to the bacterial colony being tested. And this entire purification process is what is depicted here on the right of your screen. So you see that there's the lysing, where the bacterial cells are bursted open, and then there's centrifugation in order to get that supernatant, and then you have the column loading, the washing, and the eluting in order to get the purified protein. Enzyme activity was determined by monitoring the time required for the pH of a solution containing saturated CO2 in the crude or purified carbonic anhydrase mixtures to change from 8.3 to 7.3. In addition to testing overall enzyme activity, the researchers also carried out other tests with the purified and crude enzyme mixtures. One of them dealt with carbonation and the carbonic anhydrase's ability to convert carbon dioxide to its storage form of calcium carbonate. To do this, they created a solution with 10 milliliters of CO2 saturated water, 10 milliliters of 2% calcium chloride, and 1 milliliter enzyme solution. After a definite time interval, the precipitate that formed as a product of the carbonic anhydrase's reaction with carbon dioxide was obtained and analyzed under an SEM microscope for calcite, which is the rock-forming mineral of calcium carbonate. The amount of calcite was later compared to the amount formed when other types of carbonic anhydrase bacteria were tested in other research experiments. And the picture right here on the right shows you what those SEM or the SEM microscope, the calcite pictures that are able to be formed from carbon sequestration of CO2.
Okay, so for identification of the bacteria, next they needed to actually complete a biochemical and molecular characterization of the bacteria in order to identify it. After many bacteriological tests such as gram staining, iodine tests, oxidase tests, and fermentation tests, they identified the microbe as gram-negative, rod-shaped, fermental, and acid-producing bacteria in the presence of glucose, sucrose, maltose, and lactose. After further gene sequencing and nucleotide comparisons, they decided that the bacterium that they cultivated from the soil samples was Citrobacter fornuti, which can be seen in the picture here on the right.
The results of the testing with carbonic anhydrase proved that this enzyme is affected by carbon and nitrogen sources, as well as pH and temperature. As observed in these graphs on the right, the presence of glucose and sucrose, which are carbon sources, and soybean meal, which is a nitrogen source, produce the most biomass. These conditions also coincided with an enhancement in the production and activity of the carbonic anhydrase enzyme in this bacterium. In addition, the optimal pH and temperature for carbonic anhydrase production was found at a pH of 7.0 and 37 degrees Celsius. Finally, the most important of the results in terms of carbon sequestration and the reduction of atmospheric CO2 levels was that both the crude and purified enzymes of Citrobacter fruendi have the potential to form calcium carbonate much faster than other prominent bacteria that have been tested for carbonic anhydrase activity. The purified carbonic anhydrase aided deposition of calcite was 230 milligrams of calcium carbonate per milligram as compared to crude enzyme, which is 50 milligrams of calcium carbonate per milliliter of enzyme. These both were much higher than the partially purified carbonic anhydrase isolates from Pseudomonas fragile, Bacillus pumilus, and Micrococcus lilae, which were 27.33, 25.43, and 24.02 milligrams of calcium carbonate per milligram of enzyme, respectively. Similarly, the intercellular carbonic anhydrase activity by Cytobacter fruendi was also higher than in other studies of bacterium with carbonic anhydrase activity, such as tests with Bacillus megaterium, Bacillus subtilis, and Bacillus simplex. Cytobacter fruendi had an intercellular carbonic anhydrase rate of 11.3 enzyme units per milliliter, while these other bacteria had rates of 5.61, 4.45, and 4.26 enzymes per milliliter, respectively.
Okay, and to wrap it up here, based on the test performed in the scientific article, the Citrobacter fruendi bacterium for the Himalayas plays a key role in carbon sequestration and the reduction of carbon dioxide from the atmosphere. Its carbonic anhydrase enzymes sequestrate carbon dioxide to calcium carbonate minerals at a faster and more productive rate than the other conventional microbes used. Because of this, this bacterium could be a great tool to mitigate the effects of climate change. Combined with other forms of carbon reduction, such as terrestrial and oceanic sequestration and renewable energy, this source of biological sequestration can help to reduce the amount of atmospheric carbon dioxide and stop the sun's heat from being trapped in the atmosphere. The future of climate change in terms of biomitigation lies in the hands of microbiologists. It is imperative that they come up with a way to manipulate these types of bacterium and cultivate them in a safe and organized way in order to use their sequestration abilities most effectively to combat climate change.
And this is our works cited page with the works that we used to do this presentation.